Hello, I hope you are fine and doing well. Here we have to talk microbial anatomy and physiology. If you remember, in our previous session, we were talking about the different environmental factors affecting microbial growth on this planet and the different ecological needs where environmental factors are responsible to maintain and to manage a microbial community structure and function. But during this talk, we are able to say that there is no ecological niche on this globe, and there is no environment where microorganisms are not present. But we are able to say that microorganisms are present everywhere on this globe. Either that is ocean, either that is those are glaciers, those are rocks, those are forests, that is sand, that is fresh water, whatsoever is there, but microbial populations are present there. Even microorganisms are present in oceanic benthic zones at bottom of the oceans where huge pressure, darkness, very cold, and nutrient, no oxygen, but microorganisms are present over there. If you remember, we have talked various factors such as osmotic effect on microbial growth, temperature effect on microbial growth, their mechanisms, pH effect on microbial growth, and mechanisms to maintain the microbes to their life at different concentrations. Today we have to talk oxygen and microorganisms. Okay, we are there are several types of microorganisms, and on basis of the requirement of oxygen, microorganisms has been divided into different groups. Number one, aerobes. Those the group of microorganisms which require oxygen to live, to survive, to grow, to increase their population. Microorganisms which need oxygen for to live, to grow, to proliferate, to function, those are called aerobes. In another way, we can say that aerobic microorganisms are those microorganisms which have ability to grow in presence of oxygen. If oxygen is removed, they cannot grow, they cannot live alive. So the group of microorganisms which are growing in presence of oxygen, that those are called aerobic microorganisms. So we can say the microorganisms which are present on the surface of the planet where oxygen is present. So mostly microorganisms are aerobic because there is no ecological need on surface of the globe where oxygen is not present. But there are various places where oxygen is not present, so those places are called anaerobic environment. Anaerobic places, several uh, places which are hidden in depth and they have no oxygen, so those will be called anaerobes. So anaerobes, the group of microorganisms, they don't require oxygen and may even be killed by exposure to oxygen. The group of microorganisms which can grow in absence of the oxygen, those are called anaerobic microorganisms. If those microorganisms have exposure with oxygen, either they are converting themselves into spore form, into resting structures, are going to die. So the globe where oxygen is not present means microorganisms are there and they have ability to grow in absence of oxygen, and those microorganisms are called anaerobic microorganisms. Facultative microorganisms can live with or without oxygen. The group of microorganisms which have ability to grow, to proliferate, to function in presence of oxygen as well as in absence of oxygen are called facultative microorganisms. Aerotolerant and aerobes, a group of microorganisms which are called aerotolerant and aerobes. From this word, it is clear that aerotolerant, which can tolerate the presence of oxygen, but those are anaerobes, can tolerate oxygen and can grow in its presence even though they cannot use it. So the group of microorganisms, those can grow in presence of oxygen even if they are not using oxygen, but they can grow and basically they are anaerobic. So such type of microorganisms are called aerotolerant anaerobes. Microaerophilic, a group of microorganisms can use oxygen only when it is present at level reduced from that in air. Those are called microaerophilic, means those microorganisms which can use oxygen in a small quantity if available, okay. If not available, they not need it. So those are called microaerophilic. Oxygen and microorganisms. Thioglycolate broth is the medium where we can grow microorganisms on the basis of the oxygen concentration. This is complex medium that separate microbes based on oxygen requirement. Thioglycolate is the medium which can be used for the growth of the microorganisms on the based on the oxygen requirement. This thioglycolate reacts with oxygen, so oxygen can only penetrate the top of the tube. So for the growth of the anaerobic microorganism, we can use thioglycolate broth. It is a complex medium that separates the microorganisms on the basis of the oxygen. Microorganisms which require oxygen, they will grow on top of the medium. Microorganisms that require little amount of the oxygen, they will grow beneath the surface, beneath the top, because thioglycolate reacts with oxygen and reduces its concentration. So if you will see in A, aerobic aerophilic microorganisms, they will grow on surface, and anaerobic microorganism will grow at bottom where oxygen is not present, and microaerophilic, they will grow on beneath this. These will be the microaerophilic. This is oxygen zone. Here aerobic microorganisms will grow. These are aerobic. Okay. This is anaerobic zone. Here oxygen is not present. So these may be little bit microaerophilic. They will grow over there. On the basis of oxygen, requirement, we can grow microorganisms on this thioglycolate medium. Because this is thioglycolate medium, aerobic microorganism will grow here and anaerobic will grow on down. Special techniques are needed to grow anaerobic and anaerobic microorganisms. Reducing agent, chemicals that may be added to culture media to reduce oxygen. Suppose thioglycolate. Thioglycolate is reagent which can be used in different media where they will reduce the oxygen concentration and anaerobic microorganisms will grow over there. So such type of techniques also we can use this. It will grow here or it will grow here. So here we have talked that how we can grow microorganisms without oxygen. So here if you will see, this is an aerobic incubator, or we can say carbon dioxide incubator, where these are the places from where we can insert our hands and we can put cultured plates over here, and from here we are going to switch on the uh, carbon dioxide, a small amount of carbon dioxide, oxygen, which will reduce the concentration of oxygen from this chamber, and all plates which are present over here, those will grow, microbial communities will grow here anaerobically. And this gas park plus also we can place inside of this jar. This plus this jar we will cut from here, and it will absorb all oxygen which is present inside of the container. And here environment will be anaerobic. And if in culturing media microorganisms are anaerobic, they will grow, neither they will not grow. If oxygen will be there, they will not grow. So for the growth of the anaerobic organisms, we can use thioglycolate medium, as well as we can use such type of jars where we can keep uh, oxygen absorbers, which will reduce oxygen and will allow to grow the anaerobic microorganism, as well as we can use anaerobic chambers or anaerobic incubators, where facility of oxygen reducing facility is present, or addition of the carbon dioxide facility is able, and we are putting all plates which we have to, where we have to grow anaerobic microorganisms, so it will facilitate for the growth of the microorganism. So for this, if you will see that to grow the microorganisms in unusual condition, it is very difficult. It is quite difficult, because in normal condition, in normal laboratory, we cannot grow anaerobic microorganisms. And specific special type of equipment and chemical reagents are required for the growth of the different microorganisms. So in this session, what we have studied, we are going to little bit summarize over here. If you will go back from here, we have studied different cell wall bacterial cell division. Here we have studied bacterial cell division, and where it was stated from here that cell will be elongate, cell will be septum will form, and after completion of the septum, cell will be divided into two, and these mechanisms and responsible proteins and enzymes which are responsible for this has been studied here. FTS proteins, which is a complex structure responsible for the division of the bacterial cell, and there are several proteins which are called min proteins, which are functioning for the cell to cell division. MREB proteins, which are responsible for the morphology of the bacterial cells, as well as for elongation. MREB proteins, which is responsible for the elongate of the bacterial cell by putting, we must have to make it clear, by putting uh, N-acetylmuramic acid and N-acetyl glucose amine in newly growing sites of the bacterial cell wall, which is responsible to elongate the bacterial cell. This mechanism we have studied over here, and we have studied growth curve in detail, and it was studied, and microbial growth cycle, and it is phases like phase, exponential phase, and stationary phase we have studied well, death phase as well as, and at last we have studied different factors, effect of temperature on microbial growth, as well as we have studied microbial life in the cold, microbial life in high temperature. We have studied these things in detail. Okay, temperature. And microbial in cold temperature, how microbes are getting adaptation we have studied in detail. You must have to go throughout these slides and lectures, as well as how microbes maintaining a high temperature. We have studied this thing, effect of pH, other environmental factors affecting bacterial growth, acidic pH, osmotic pressure, oxygen, and toxic forms of oxygen we have also studied. So for the detail of the study of this topic, you can go throughout any book, and you can go throughout Lexar or throughout Prescott, where these all factors has been discussed in detail. Either you can talk with your colleagues, with your friends, you can go through research papers, and you can understand this mechanism in detail, that how microorganisms are affected by different environmental factors, because different environmental factors are responsible to select a microbial community in any ecological need. Suppose there is a forest, in forest there is a specific microbial community, and that microbial community has been specified by the chemicals which are releasing from the roots of that forest, and the temperature of that forest, the moisture content of that forest, as well as pH of that humus, and other chemicals which are releasing from the root of the plants, those are responsible to specify the microbial community structure and its function. When this community is designed and defined by environmental factors in an ecological niche, if any other species tries to invade into that ecological system where environmental factors are specific, first of all, environmental factors restrict the growth of that particular invading microbial species, which is unable and not capable to survive, to grow, and to proliferate over there. Secondly, the existing microbial communities which have been developed from decades and several from long time over there, they are releasing certain chemicals which are killing to the invading microbial community into that environment. So we have also studied how microbial communities are uh, sustaining or managing to the changing environmental conditions in environment. They have different mechanisms according to the environmental conditions. So microorganisms are present on this globe everywhere, either that is extreme cold temperature, either that is extreme hot temperature, either that is extreme alkaline temperature, or extreme uh, acidic pH. Alkaline pH or acidic pH. Microbes are present everywhere. So microbiology person have ability to work in every type of environment where microbes are present. Therefore, we are saying that microbiology persons have ability to work throughout the globe where microbes are existing. So thank you for your attention, and I hope you will study it later more by going later.